Good day, everyone. I will be discussing the hematopoietic system as an overview of our hematology module. Now, you say hematopoietic. Hemato means blood cell. Okay. The word hema means blood. Okay. And then poietic means formation. So when we talk about hematopoietic system, we're actually talking about how the different blood components, the blood cells, how these blood cells are formed. No? So hematopoiesis is the normal formation and development of blood cells in the bone marrow. It's actually a continuous regulated process of blood cell production that includes renewal, proliferation, differentiation, and maturation. Now, I'd like to put emphasis that hematopoiesis is actually a normal, okay? It's a normal process that has been happening in the body, no, particularly in the bone marrow, because we actually have instances wherein there is abnormal formation and development of blood cells in the bone marrow that may likely produce disease symptom to a human. No? For example, say leukemia. In leukemia, leu means white, no? Leukemia, there's actually increased white blood cell production. And this is actually not normal anymore. Now, hematopoiesis being a normal, okay, being a normal process of formation of blood cells in the bone marrow is actually regulated. Regulated in a sense that there are actually different mechanisms, feedback mechanisms, and different factors, growth factors, for example, which will actually help regulate in the process of blood cell production. Say, for example, in red blood cell production, no? Red blood cell, the main regulatory process that would actually induce formation of red blood cell is hypoxia. So if the cells are exposed to decreased oxygen tension, chances are your kidneys will produce a factor, a growth factor, a substance that will regulate the process of red blood cell formation. And that substance is what we call erythropoietin. That's just an example. In platelets, no, we have in cases wherein there is decreased production or um, increased platelet destruction, okay, the organs in the body that are capable of, are some organs in the body are capable of producing a substance that will regulate production of platelets. And we call this substance thrombopoietin. In in case of WBC production, we call it leukopoietin. Okay. Now, the main purpose of this hematopoiesis is to actually replenish. Okay. Remember, we continually have turnover. Okay. We have normal turnover of our blood cells in the body through the reticuloendothelial system or your RES, meaning your cells age. As they age, they die, and your body removes the senescent, okay, senescent population of cells because they are non-functional anymore. And so to renew, to replenish that population that was turned over, that was removed, no? This hematopoiesis is a process of renewal. Okay? And then proliferation in cases wherein there is actually for example, in immune response, okay? Say, for example, the T cells and B cells, particularly T cells. When the body is exposed to an antigen, when there is an antigenic challenge, there is actually a need for the immune system to proliferate the immune cells to hasten removal of the antigen, okay, which has elicited the immune response. And so the role also of hematopoiesis is for proliferation of cells, no, to increase cell number. And then differentiation and maturation. No, differentiation in a sense that, and maturation in a sense that, initially being a process, no? And this process is actually developmental, which means the entire process begins by producing an immature cell, and that cell eventually matures. Okay, so we start with a stem cell, and then these stem cells, as it matures now, it produces a more mature type of cell capable of different type of function. Now, generally, your hematopoiesis has three developmental phases. We have this what we call mesoblastic phase, hepatic phase, and myeloid phase. You say mesoblastic, this basically occurs in the yolk sac, no? During your fetal life. This happens during fetal life. And when the fetus has developed liver, okay, what follows is what we call hepatic phase. Hepatic because main production of blood cells occur in the liver. And when there is now formation of bone marrow, okay, bone formation and then bone marrow formation, hepatic phase is followed by the myeloid phase. So let's start with mesoblastic phase. As I've said, this occurs in the yolk sac, and so this is also termed as the yolk sac phase. Primitive because this occurs during fetal life. This is actually the first round of hematopoiesis that occurs during fetal life. Now, usually in early fetal life, no, the first event that happens is blood production in the blood islands, no, which develops in the mesoderm of the yolk sac. The key word here is formation of blood islands in the mesoderm of the yolk sac. And this usually occurs as early as 19 to 20 days of gestation. No? 19 to 20 days of life. And this continues up until 8 to 12 weeks of gestation. In the yolk sac phase, okay, the cells that are initially produced are what we call progenitor cells. And these progenitor cells will give rise to hematopoietic stem cells. These hematopoietic stem cells, as I have mentioned, since hematopoiesis is a developmental process, these hematopoietic stem cells will eventually mature to perform separate sets of function, different from those function performed by your stem cells. So in other words, okay, the mother cells, no, the cells that will eventually mature to become blood cells are actually your stem cells, no, which are derived from certain progenitor cells in the aorta gonadomesonephros region of the yolk sac. So these are derived from the yolk sac region. Now, during this phase, okay, there is initial production of your red blood cells. No, we call it erythropoiesis. Poiesis meaning formation, erythro meaning red. Okay, red blood cell. So erythropoiesis means red blood cell production. And some areas of white blood cell production and platelet production. Now, since erythropoiesis starts to occur in yolk sac phase, okay, it is understood that, remember, your red blood cells contains the protein hemoglobin, particularly globin. And globin is complex with a heme compound to form hemoglobin, which is responsible for the oxygen carrying function or capacity of your red blood cell. Now, since there is initial red blood cell production in the yolk sac phase, it is understood, we can imply that there is also globin chain production, because you cannot actually form a red blood cell if you don't have these precursors, the globin and the heme. And for examination purposes, take note of the different embryonic types of hemoglobin that occurs during the yolk sac phase. So we have embryonic, huh? We have gower 1, gower 2, and Portland hemoglobin. Okay. Then we have hepatic phase, no? We call it definitive hematopoiesis. Definitive hematopoiesis because this is where the other forms of blood cells, no, begin to develop, nor they are formed, no, not just red blood cell compared to the yolk sac phase. There is also production of your myeloid cells, no, particularly your granulocytes, and lymphoid cells, also your lymphocytes. Okay, so hepatic period, from the word hepatic, main production or the main site of hematopoiesis occurs in the liver. And this usually starts at the 5th to 6th week of gestation. Okay? 5th to 6th week of gestation, and this continues between 10th and 30 weeks of gestation. It could also remain active until the 1st and 2nd week after the child was born. And the liver, okay? The liver being the primary organ in primary hematopoietic organ in the hepatic phase or definitive hematopoiesis phase, okay? It is capable of producing your red blood cells, your granulocytes, and your lymphocytes. Aside from the liver, your spleen also, the thymus, and the lymph nodes become active also in red blood cell production. Okay? So majority of the erythroid cells, no? Erythroid cells are cells which become mature red blood cells, no? Erythroid, okay, are produced in the fetal liver. But some, okay, some organs contribute to the production of granulocytes and lymphocytes, that is your spleen, thymus, and lymph nodes. So at this point, the cells are morphologically identifiable. Different with the yolk sac phase, because here, your hematopoietic stem cells and progenitor cells are actually non-identifiable, meaning you could not tell that this cell is actually a stem cell and that this cell will actually develop into a red blood cell, a leukocyte, a white blood cell, or a platelet, because these hematopoietic stem cells and progenitor cells are actually morphologically non-identifiable unless you perform markers. No, you test for the different surface markers, then you can say this is a stem cell, this is a progenitor cell. But under the microscope, it's really difficult to know that this is a stem cell or not. But when hematopoiesis enters hepatic period, you can actually differentiate, no, the lineage, no, as to if it is in the erythroid, okay, erythroid lineage, myeloid lineage, or monocytic lineage, even lymphoid, no, you can differentiate. Then lastly, we have your medullary or myeloid period. The key word here is that the main site, no, the primary site of hematopoiesis is in the bone marrow. And this starts in the 5th month of gestation. Okay. So bone marrow cell production during 5th month of gestation. And what's really highlighted during the medullary period, that's why this is also called myeloid period, is because there is predominant production of your myeloid cells. Now, myeloid cells are actually your white blood cells, okay, that will most likely develop into neutrophils, eosinophils, basophils, monocytes, no? Different from lymphoid. Lymphoid will it's actually having a separate mother cells or lineage. No, we call it also lymphoid, no, lymphoid cell. That's why we'll have different discussion. You'll see in the next few slides. Okay, but for this particular slide, I want you to know that this happens during the 5th month of gestation in the bone marrow, and the predominant cells that are produced are myeloid cells. Okay, there is also production of hemoglobin A1, no? Hemoglobin type A1 consisting of 2 alpha and 2 beta globin chains. So these are actually the post-fetal life type of hemoglobin. We'll have a separate lecture for this. So various stages of maturation in all 3 lineages can be seen. So what are these lineages again? Erythroid for red blood cell production, myeloid for WBC production, for granulocyte production, and lymphoid, okay, for lymphocyte production, or different lineages. So this diagram would actually highlight, no, the different times. This is actually a diagram showing us the time of maturation, okay, or maturation of cells and the primary site of hematopoiesis, such that again it begins in yolk sac, followed by hepatic phase, and then eventually fifth fifth month of gestation, no, bone marrow takes over, okay, in the myeloid phase, and in adults, okay, so bone marrow is also the main site of hematopoiesis, no, particularly these bones. Okay. So this is now the lineages that I've been telling about. Remember, production of your progenitor cells and stem cells occur in what phase again? Okay, they occur in the yolk sac phase. Okay, and then these progenitor cells become, no, they mature and develop into hematopoietic stem cells. And these hematopoietic stem cells is responsible now for giving rise to different types of cells, so depending on the stimulus. Okay? So, the hematopoietic stem cell matures into either common myeloid progenitor and common lymphoid progenitor. You say myeloid because it gives rise to granulocytes, no? And then common because all granulocytes, no, even erythrocytes have will develop from this common progenitor cell. Then lymphoid because it gives rise to lymphoid cells, no, particularly mature lymphocytes. So let's run through these diagram. So under common myeloid progenitor cell, depending on the stimulus or the growth factor that acts upon, okay, the common myeloid progenitor cell, it may give rise to a granulocyte monocyte progenitor. Granulocyte because it may give rise to a granulocyte. Okay, for example, a neutrophil, an eosinophil, or a basophil. Okay, this is actually different, no? Different na lineage. Okay, then monocyte because it gives rise to a monocyte, which eventually matures into a macrophage. Then we have eosinophil, basophil, progenitor. Eosinophil, basophil, progenitor because it gives rise to a mature eosinophil, basophil because it gives rise to a mature basophil. So for examination purposes, you really need to memorize the different stages of maturation. Okay, starting with the multipotent progenitor stem cell, going to progenitor and then the precursor cells and then the mature cells in the blood and in the tissue. So from the most immature, no, myeloblast, Myeloblast matures into promyelocyte. Promyelocyte matures into myelocyte, then to metamyelocyte, then to band, and then to segmented neutrophil. So we will discuss this individually in the next few slides. Then monoblast matures into a promonocyte into a monocyte, and then in the tissue, when monocyte migrates into the tissues, it becomes now a macrophage. Then, more or less the same name, no, the same maturation series. However, just add the term eosinophilic for eosinophils, basophilic for basophils. Now, there's actually a different nomenclature for the erythroid series. No, we call this the granulocyte series, the monocyte series. Since eosinophil and basophil actually have more or less the same nomenclature, so we can also call these two as granulocytic series. Okay, so there's actually a different nomenclature for erythroid series. So we say erythroid, this is actually the series that will the series or maturation series that will lead to the formation or development of immature erythrocyte. Okay, so we start off with a pronormoblast or an erythroblast. Okay, then basophilic normoblast, polychromatophilic normoblast, orthochromatophilic normoblast, PM or polychromatophilic macrocyte or erythrocyte or reticulocyte, and then erythrocyte. No? Notice that as the cell matures, it eventually loses its nucleus. And so a mature red blood cell do not contain nucleus. Now the same mother, no, the same progenitor cell is your megakaryoblast, okay, which becomes now matures into a pro-megakaryocyte and eventually into a megakaryocyte. We actually have meta-megakaryocyte after this megakaryocyte, but actually very difficult to find in slide or SMEs. No? Platelets are actually not cells. They are not cells. Okay, platelets are actually fragments of a megakaryocyte cytoplasm. So the most mature stage, no, of maturation is your megakaryocyte. Okay, platelets are not cells. Then under common lymphoid progenitor, so the mature forms of lymphoid cells are actually T and your B cells. Okay, so we have a pro B cell, a pre B cell, and then a pre T cell, which matures into a lymphoblast, and then this lymphoblast will mature into a B or T lymphocyte. The B lymphocyte, it goes into tissues, it becomes now plasma cell. Then aside from the T and the B cells, we have dendritic cell, which is also which developed from lymphoid progenitor cell, and also your NK cells or your natural killer cells. Okay, so this is another diagram showing real pictures. Okay, real pictures of the different cells that I have mentioned. No worries, because we will discuss this one by one. So let's start off with the different cells of the myeloid series. Okay, what are these cells of the myeloid series? So we start with the most immature one. We term it as myeloblast. Okay, so you can pause this video lecture and try to memorize and recall the different cells, no, in the myeloid series. Okay, memorize first the series and then go back to this slide. Go back to this, play the lecture video again. All right. Now let's start with myeloblast. This is actually the most immature cell in the myeloid series. Now, we actually have in the laboratory, there is actually a need to identify immature from mature cells, because presence of immature cells would actually tell us a different story. Okay, for example, if you have a lot of myeloblasts in the bone marrow and in your blood circulation, that is actually indicative of leukemia. Okay, now you really have to understand that there is a need to properly identify these immature cells because, as I've mentioned, this is indicative. Increased numbers of these immature cells in the blood circulation is indicative of a disease process. Okay? Now, let's identify the cells. Now, focus first on the picture. Before you read anything here, focus first on the picture. I will help you identify the cells. Now, let's start by identifying the structures inside and the structures outside the cell, no, outside the nucleus. No, the main structure inside is actually a nucleus, and then the one surrounding it is the cytoplasm. Okay, so let's start off with the size of the cell. Basically, when we talk about a blast, no, or an immature cell, any immature cell is actually large, actually a big cell. Okay, now as the cell matures, the size of the cell becomes smaller. Okay, so first identifying criteria there is the size. A blast or an immature cell is actually a big cell. Okay, now we go to the nucleus of the cell. Look at the nucleus of the cell. Look at the staining pattern. Notice that it is colored predominantly blue to violet. In this case, no, that's the stain, H&E stain, right stain. This is right stain rather. Okay, it's actually colored violet. Okay, meaning if it's colored violet, it took up the basic dye. Why do you think? Remember, if you can still recall the basic, no, basic principle, basic organelles, the nucleus will actually contain the genetic material DNA and RNA. And these DNA and RNA are actually ribonucleic acids, deoxyribonucleic acids. No, they are acids. So they are acid. Most likely they are to take up the dye, the part of the stain, no, the dye found in the stain, which has the opposite charge for an acid. No, that is now the basic dye. That's why the basic dye being colored blue-violet, blue to violet in range, the nucleus will stain now violet. Now, the more, okay, look at now the let's look at the details inside the nucleus. As you can see, the entire nucleus is stained homogeneously. Homogeneous meaning all parts of the nucleus actually takes up equal amounts of the dye. No part is darker, no part is lighter, okay, except for the nucleolus, of course, okay, meaning the entire nucleus is homogeneously stained. It takes up equal amount of the basic dye. This means that, okay, this means that the more homogeneous looking a cell nucleus is, that would tell us that the cell is more immature. Okay? Compare it to the next cell. You see the next cell. As the cell matures, you get to see non-homogeneous staining pattern. So some part is dark while some part is lighter. No, compared to this one, everything is stained homogeneously. It appears smooth and homogeneous. Now, we generally term it as the chromatin pattern of the cell. Now, the chromatin pattern of the cell is very delicate. Delicate in a sense that it is very fine. And so, if it is fine, meaning fine, no, very nipis, very fine, it would only take up a small amount of the stain. Compared to a more mature cell, no, as the cell matures, the chromatin becomes dense and plumped, and so it takes up a lot of the stain, and so it appears darker. Compared to this, since the chromatin pattern is really fine and delicate, it is actually evenly stained, no, very smoothly and evenly stained. Now, that is your main determinant, no, that this cell is immature. Another determinant is the presence of nucleoli or nucleolus. Nucleolus is actually the lighter stained area of the nucleus, no, nucleolus, two or more distinct nucleoli or nucleolus. Now, presence of this lighter stained area or nucleolus would tell us that the cell is immature. It's a blast. Okay? Take note of that. Nucleolus. Now, another distinguishing feature, okay? So we have the chromatin pattern, very smoothly, evenly stained, fine chromatin pattern, the presence of nucleolus. Another thing is the nucleus is so big, okay? The nucleus is so big that it occupies most area of the cytoplasm. Thus, if the nucleus is so big, occupying much area of the cytoplasm, the nucleus to cytoplasmic ratio is very high. Meaning, the cytoplasm now, volume is actually lower as compared to the nucleus, nuclear volume. And the normal volume NC ratio, no, of a blast or an immature cell is 7:1 to 5:1. My point here is that an immature cell or a blast should have a big nucleus, bigger NC ratio. Okay? Now look at the cytoplasm of the cell. Now the cytoplasm of the cell, okay, if you can look at it, it's really smooth and usually non-granular. You could not see any ribosomal granules, no? Very smooth, no granules at all. And the cytoplasm is blue stained also, no? Blue stained meaning if it's blue stained, what can we infer? It actually contains genetic material. So a lot of genetic material, particularly RNA. Okay? Because this cell is actively dividing. All right. So again, the main distinguishing feature of your myeloblast is the presence of a smooth chromatin pattern, presence of nucleolus and stained area, no? And then the cytoplasm is non-granular and with, of course, a high NC ratio. Then, as the myeloblast matures, no, the next cell in the series, maturation series, is your promyelocyte, also known as progranulocyte. It's called progranulocyte because in contrast to your myeloblast, you can now see the granules in the cytoplasm. Remember, a myeloblast has no granules in the cytoplasm. But a progranulocyte, as you can see, these dark stains here are actually granules. Okay? These are called primary granules. Okay, these are dark blue or reddish blue primary or azurophilic granules. Okay? Now let's examine the nucleus. Again, I've mentioned that as the cell matures, the size of the cell decreases and the nuclear size also decreases, the cytoplasmic size decreases, and so comparing it here, you see, no? Almost the nucleus is covering the entire cytoplasm. But here, we have a smaller nuclear size with more or less larger, no? Larger cytoplasmic size. However, the NC ratio is still high. But let's examine the nucleus first. No, look at the nucleus of the cell. Okay? The nucleus of the cell, as you can see, is not homogeneously stained anymore. Compared it here, it's homogeneously stained, no? Homogeneous meaning all parts of the nucleus takes up evenly the stain. But in here, we actually see parts which are, some parts are darkly stained, no? Look at this part here, very darkly stained. Some parts are lightly stained, meaning the staining pattern of the nucleus is not even. Okay, compared it here, it's really smooth and even. This is because as the cell matures, as I mentioned already, no, as the cell matures, the chromatin pattern is slightly coarser. Magkabaga siya, no? Magkabaga, it becomes thicker. The chromatin compared to here, that the chromatin is really fine and delicate, no? That's why it's smoothly stained. So if baga siya, if it's thicker, chances are it will take up a lot of the dye or the stain. And so it becomes more darkly stained compared to your myeloblast. So it's darkly stained. It has some darkly stained areas due to a slightly coarser chromatin pattern. And the distinguishing feature here, it still retains your nucleoli or nucleolus. Okay. The main distinguishing feature, if you see a cell, no, if you see a cell like this, and then you're really confused as to if it is myeloblast or promyelocyte, the main distinguishing feature here is the presence of granules in promyelocyte. Presence of granules. If you do not know what a chromatin pattern is, Doc, it's really difficult to see, Doc, nga this area is stained darkly and the other cell has finely stained area. Well, look at the cytoplasm. Okay, presence of granules. And of course, no, there should be nucleoli. There should be nucleoli because the next cell also contains granules in the cytoplasm. Now, the next stage of maturation is what we call myelocyte. If it is bound to mature into a neutrophil, it's called neutrophilic myelocyte. If it is bound to mature into an eosinophil, it's termed as eosinophilic myelocyte. If it's bound to mature to a basophil, it's termed as a basophilic myelocyte. Now, let's examine the cell closely. The cytoplasm is actually abundant now. Why abundant? More abundant compared to the two cells that I have discussed. Because again, as I've mentioned, as the cell matures, the nuclear volume, okay, decreases and the cytoplasmic volume increases. So there is abundance of cytoplasm. The cytoplasm, the same with myelocyte, this is also filled with granules. But take note, a promyelocyte is filled with primary or azurophilic granules, whereas your myelocyte cytoplasm is filled with secondary, okay, secondary or specific granules. Now, we have to distinguish the two because the specific and secondary granules actually contains different substances. No? And we will discuss what are these contents in the next few lectures. Okay? But for now, I want you to remember that the granules in the cytoplasm of a promyelocyte is called primary P. Pro for P for primary azurophilic granules, or in the myelocyte, these are specific or secondary granules. Take note. Huh? Now, look at the nucleus. Remember I mentioned that as the cell matures, what's likely to happen? The chromatin pattern thickens. So you see, no, there's actually a thicker staining pattern of the chromatin because this cell is more mature. Compare it with your promyelocyte, although there's some areas here which are thick, but it's still thinner compared to this one. And then you look at the myeloblast, no? Very fine. No thick strands, no thick areas here. That we can say that is thick, that is fine because of the amount of stain that it takes up. Okay? Such that the darker the area of the nucleus, meaning the thicker the chromatin is in that area. But in here, look at the cell. There's actually no thick areas, no darkly stained areas. That means that the chromatin strands in a myeloblast are fine and very delicate. No? Compare it here in myelocyte. the chromatin is actually thicker. Okay? Thicker chromatin pattern. And how, Doc? It's difficult for me, Doc, to know. I know nga, this has granules. It could be a promyelocyte or a myelocyte. But it's really difficult for me to say this is thick chromatin or not. Okay? Another distinguishing feature of your myelocyte is absence of nucleolus. If you see a cell with granules, thick chromatin pattern with nucleolus, then it's a promyelocyte. You see the nucleolus? But absence of nucleolus would tell us that it's a myelocyte. Okay? Another distinguishing feature of your myelocyte is the flattened D shape of the nucleus. No, it's flattened on one side and it's kind of bulging, no, on the other side. More or less, it looks like letter D. It's flattened on one side. Okay, take note of that. Another, again, look at the cytoplasm, the nucleus, the nucleoli, no, scallop appearance. This one, the granules, etc. Okay. Let's proceed to metamyelocyte. So remember, I mentioned that in myelocyte, the one side of the nucleus starts to flatten. Now, eventually, this side starts to form invaginations. And once you see an indention here, we term it now as metamyelocyte, provided that the indention is less than 50% of the total diameter of the cell nucleus. Okay? So as the cell matures, the cell size decreases, the nuclear size decreases, the cytoplasmic volume increases. Okay? Now, your metamyelocyte, or also known as juvenile cells, okay, contains granules in the cytoplasm, both primary and secondary granules. But the main distinguishing feature here is the presence of indentation. That's why we also call this as a kidney or bean-shaped nucleus. Okay, a cell with a kidney or bean-shaped nucleus because of the indention. Provided, again, that the indention must be less than 50% or less than 1/2 of the diameter of the round nucleus. Because if the indention is more than 50%, okay, we call this cell now as band or stab or staff. So notice the indention is more than 50%. But here, the indention is just less than 50%. Okay? So the same identifying markers, a more clumped chromatin pattern, absence of nucleolus, and presence of granules in the cytoplasm. But a distinguishing feature is the indention here. For band, look at the chromatin pattern is more clumped, no? As the cell matures, and more indention. This time the indention is more than 50%, or more than 1/2 the diameter of the round nucleus. Okay. And then look at the granules. No, it's called granulocytic series for a reason. It's granulocytic series because of the presence of granules. Now, if you see, fine azurophilic or purple, light violet purple granules, most likely this cell will develop into a neutrophil. But if you see a granules which are orange, no, red-orange granules, bigger granules, most likely this cell is going to mature into an eosinophil. But if the granules is blue-black, most likely it will mature into a basophil. Okay? So, segmented neutrophil, once you see fine threads of segments, that cell now is called segmented neutrophil. Now, to differentiate it with a band does not form segments, no? You see, it forms an indentation, but there's no segments present. But if you see segments, a thread-like segments like this, that is now considered the most mature cell in the maturation series. No, we term it as segmented neutrophil. Okay, so notice that a segmented neutrophil has been segmented or separated into 2 to 5 lobes. Take note of 2 to 5 lobes. Some books would say 3 to 5 lobes. There is actually a significance of the number of lobes, because if the lobes is more than 5, we call that cell now hypersegmented neutrophil. Or if the cell now has less than 2 lobes, okay, we call it now 2 or less lobes, we call it now hyposegmented neutrophil. Hypersegmented neutrophil actually has a clinical significance, no? For example, in megaloblastic anemia. No need to really dwell on that, no, because we'll have a separate lecture on that. Hyposegmented in Pelger-Eway anomaly, sorry. Pelger-Eway anomaly. Pelger, Pelger-Eway, HUET anomaly. Okay, so no need to memorize on that for now. But what I'm trying to point out is that the number of lobes should be within this range. No? More than 5, hypersegmented. 2 or less, take note, 2 or less, hyposegmented. Okay? The normal value is there. Now, take note of the appearance. No? The main differentiating feature here is the characteristic granules. Again, if it's fine, blue-violet granules, we classify the cell as neutrophil. But if the granule is red-orange, bigger granules, we term it as eosinophils. Now take note, there is a need to really look at the granules, because the granules are the only differentiating feature between a neutrophil, immature neutrophil, immature eosinophil, and immature basophil. Look at the granules, please. Okay, look at the granules. Bright orange, pink granules. Now, usually, the clinical significance for neutrophils is that this type of cells are usually increased in bacterial infection. Okay, bacterial infection, such that when you look at your, if you happen to look at a CBC result, no, if you look at a CBC result, then you see segmenters or neutrophils there. If it's more than 50 to 70%, most likely the patient has bacterial infection. But if eosinophils is actually increased, no more than 1 to 5%, most likely this is associated with conditions such as parasitic infections, no, helminths infections, and allergic reactions, no, eosinophils. Then, if the granules in the cytoplasm are blue-black in color, the cell now is classified as basophil. Okay? So usually basophil is seen in chronic myelogenous leukemia, no? CML, chronic myelogenous leukemia, myelogenous, myelogenous leukemia. Also seen, no, in anaphylactic reactions. Okay? And then also seen in conditions wherein, aside from chronic myelogenous leukemia, no, this is also seen in conditions wherein the patient has been exposed to really chronic allergens. Okay? But for allergic reactions, if you are asked, you answer eosinophils. So notice also that the granules here, you see, no, some cells have a lot of granules, some cells have only a few granules. Take note that the purpose of this, the reason of this is that these granules are water soluble. So this cell containing only small amounts of granules, maybe during the staining process, the granules were dissolved, no, in the water. Okay, so the granules actually are covering the nucleus, and the color of the granules are blue-black. no, basophil. Okay. Then another cell in the granulocytic series. Okay. We have this what we call monocyte. Okay. So again, go back to the diagram of the hematopoietic series. Locate where monocyte is. Locate what is the progenitor and the stem cell of your monocyte. Now, the distinguishing feature here of your monocyte, let's start first with the nucleus, okay? The nucleus is actually variable in shape. So you can actually see some lobulations, some lobulated, some horseshoe shape, some brain-like no convolutions or foldings, very variable in shape. But what I'd like to point out is that the chromatin pattern here are actually loose, no? Loose. You see, if you compare it with a mature basophil, neutrophils, and eosinophils, look at the pattern. This actually clump, no? Clump of chromatin. Clump ang chromatin because the cell is mature. So look at the chromatin. Darkly stained, some areas lighter stained, no? Clump chromatin pattern. But it's actually different for your monocytes. Why different? It's because the chromatin is actually fine, no? The chromatin is really fine, and the term that we use to describe it is a lacy chromatin pattern. Lacy chromatin pattern. Okay? The chromatin is loose strands, okay? And it's colored light purple. Okay? Lacy chromatin pattern. Now, another distinguishing feature of your monocyte, since a monocyte is a phagocyte, remember I taught you that what happens during phagocytosis? The bacteria or the foreign organism is ingested and it forms a vacuole inside the cytoplasm. So this is now the phagolysosome. Okay? These vacuoles are phagolysosomes, meaning that this okay cell now is a phagocyte undergoing active phagocytosis, no? So presence of vacuoles in the cytoplasm or sometimes in the nucleus would most likely tell us that the cell is a phagocyte, no? Particularly a monocyte. Okay? So cytoplasm is described as ground glass appearance. Ground glass because it's actually the color actually ranges from um, light, no, to streaks of blue or violet, okay? Some grayish, okay? That's why it's called ground glass appearance. And notice also the presence of pseudopods, okay? See the pseudopods there, the tail-like extension of the cell. Then let's go now to the lymphoid series. Again, go back. I'll give you a few seconds to go back to the diagram which I have showed earlier. Pause this video and go back to the diagram. Okay. Now, lymphoid series means that the most mature cell, okay, here are actually lymphocytes. Okay. So, lymphocytes were derived from the most immature cell in the series we call lymphoblast. Take note, ha? Lymphoblast is actually different from myeloblast because your myeloblast will give rise to your myeloid cells, whereas your lymphoblast will give rise to your lymphoid cells. Now, how do we differentiate myeloblast from lymphoblast? Here, here. Notice that as I've mentioned, being a blast, no? The cell is actually larger, and then as the cell matures, it becomes smaller. Okay? Now, I want to direct your attention to the cytoplasm and the nucleus. Notice that the volume of the nucleus itself is actually more or less almost occupied. The volume of the cytoplasm rather is almost occupied by the volume of the nucleus, meaning a very high NC ratio, no? Much bigger nuclear volume compared to the cytoplasm. We term it as scanty cytoplasmic volume, meaning there's only very small amount compared to your myeloblast. If you look at myeloblast, go back to the picture, there's actually higher volume of cytoplasm. But the cytoplasmic volume of a lymphoblast is actually very smaller, no, as compared to your myeloblast. Thus, the term scanty cytoplasm. Okay? And of course, being an immature cell, being a blast, the cytoplasm is non-granular or agranular. A means non-granular and blue in appearance. Now, I want you to look at the nucleus of the cell. Okay? The nucleus, being immature, should contain nucleolus or nucleoli. And the color is actually violet, blue to violet, because of the presence of the ribonucleic acid. Now, the main differentiating factor here is very difficult to differentiate, no, lymphoblasts from myeloblasts. But if you look at the staining pattern of the chromatin, okay, look at the staining chromatin pattern. Both are homogeneously stained. But as to the intensity of the color of the chromatin staining, lymphoblast would actually appear darker because it has coarser chromatin pattern. So go back to the picture in myeloblast. Look at the picture, no? The staining of the nucleus is lighter. Compared to your lymphoblast, it's much darker because of the coarser chromatin pattern. But let's just say, Doc, it's so subjective, Doc. It's too difficult to say this one's darker, this one's lighter. Look at the cytoplasm. If the cytoplasm is scanty, then that is probably a lymphoblast. Okay? Lymphoblast. And then also, if you look at the cell itself is round, but if you go back to my picture, previous picture in myeloblast, go back to the picture now. There's actually irregular shape, no, of the cytoplasm. We call it scalloped appearance, scalloped cytoplasm, scallop because it confers to the shape of the surrounding cells. Versus the lymphoblast, it maintains the circular, no, the round shape. Okay, scallop ang myeloblast. Then it matures into a lymphocyte. So notice that the lymphocyte is a small cell. If for you to identify that this is a lymphocyte, you look at the surrounding cells. The surrounding cells are actually red blood cells, more or less the same size with a red blood cell. But how do you know that this is a lymphocyte? You look at the staining, that's the staining of the chromatin. Look at the chromatin, no? It's the chromatin is really clumped as compared to your immature cell. And look at the cytoplasm. Okay, the cytoplasm is scanty also, no, small amount of cytoplasm, colored blue, and colored blue cytoplasm. That's why we also term this cell as Rubin's. The cytoplasm, we call it Rubin's egg blue cytoplasm because the cytoplasm is very blue. So if you see a small cell, more or less the same size of a red blood cell, no, surrounding it, and the cytoplasm is very blue and scanty with clump chromatin pattern, you see the clumped, no, those darkly stained area in the nucleus, no, more likely this cell is a lymphocyte, a small cell, no, bright sky blue or the Rubin's egg blue cytoplasm. Now, sometimes you actually do not see lymphocytes which are small, okay? You see lymphocytes which are very big, and it would be very difficult for you to really identify, this is this lymphocyte. This is a lymphocyte. This is not a lymphocyte, okay? But there are actually distinguishing features. No, you just look at what I've taught you. You go back to the basic. No, I taught you start with the nucleus. So is the Is the cell mature or not? So look at the nucleus. Is the staining pattern of this cell um, mature or not? So you see clumps of cells. So most likely, this cell is a mature cell. Okay, clumps of cell. Now, you see a blue cytoplasm with some granules. Okay, blue cytoplasm with some granules and scalloped appearance of the cytoplasm and absence of nucleolus. No, so this is actually your variant lymphocyte. This means that you have an increased level of variant lymphocyte, no, because there is actually viral infection of the human, no, the where we got the blood sample. So once your lymphocyte are exposed to an antigenic challenge, no, particularly viruses, you have an increased levels of your variant lymphocyte. For example, in EBV or Epstein-Barr viral disease. Okay, but most likely, if you look at a CBC parameter and you see an increased level of lymphocytes, most likely it's pointing to a viral cause of the infection. So again, neutrophils increased level in bacterial infection, eosinophils in parasitic infections and allergic reactions, basophils in chronic myelogenous leukemia, and then your lymphocytes, no, particularly variant lymphocytes in viral infection. So the cytoplasm is abundant, blue cytoplasm, some will have granules. The cytoplasm also is scalloped. Doc, it looks like myeloblast for me. It looks like a blast. Okay, again, go back to the nucleus. The nucleus is clumped. Remember, for this cell to be considered a blast or immature cell, the nucleus must be homogeneous with no clumps, no? Namshay clumps. And then absence of nucleolus would tell us that this is a mature cell because as the cell matures, you will lose the nucleolus. Okay? Let's try this, no? Try to identify the cell. Okay, let's identify the cell. The cell, let's start first with the shape, no? It's a round cell. And then we go to the nucleus. The nucleus is clumped. So is this cell mature or not? It's more or less a mature cell, no, presence of a clumped nuclear chromatin pattern and characteristic threads, fine-like segments or threads. No, 1, 2, 3, 4. 4 segments, presence of azurophilic granules, okay, blue to violet fine granules in the cytoplasm. This is actually a neutrophil, okay, a segmented neutrophil. Next, presence of, look at the size of the cell. It's actually a cell bigger than your red blood cells. And start always with the nucleus, no? Is this cell mature or not? Look at the chromatin pattern. Clumps of chromatin, okay, in the nucleus. So is this cell mature or not? This cell is mature because of the clumps of chromatin pattern. And you see threads. So most likely this cell is segmented. But what type of segmented cell? So look at the cytoplasm. Characteristic blue, not blue, no? Characteristic red or orange granules. Most likely this is an eosinophil or segmented eosinophil. Go to letter C. Is this cell mature or not? So look at the cytoplasm, look at the nuclear pattern. The cell has more or less fine chromatin because there's less clump, but we can say that the cell is mature because of the absence of nucleolus, no nucleolus. And then look at the cytoplasm. The cytoplasm is ground glass. Ground glass means it's shades of gray and violet and blue. But your main distinguishing feature here is the presence of vacuoles, which means that this cell has been undergoing is a phagocyte. And so these vacuoles here are representative of the ingested material, no, or in the phagolysosome. So this cell having a ground glass appearance, fine lacy chromatin, with vacuoles on the cytoplasm is a monocyte. Okay? Doc, monocyte. Why not a typical or variant lymphocyte? Why not variant lymphocyte, Doc? Look at the chromatin pattern. Diba? Look at the chromatin pattern. It's actually fine, no, compared to your variant lymphocyte, which is clumped. How about this one? You see segments, no? Segmented or fine thread connecting lobes, but with a clump chromatin pattern, and then fine granules, blue-violet granules in the cytoplasm. This is most likely a segmented neutrophil. How about this? Irregular shape, nucleus with fine chromatin pattern, and then presence of vacuoles in the cytoplasm. This is still a monocyte. How about letter D? Now, letter D, actually, if you look at the cytoplasm, is an nucleus. Is this mature or not? This is mature. Why mature? Because look at the clumps of chromatin in here. Clumps of chromatin, those darkly stained areas, and the absence of nucleolus. So this is a mature cell, okay? And then we look at the cytoplasm. The cytoplasm is colored blue. Blue with abundant cytoplasm with scallop. No, scallop meaning, what do you mean by scallop again? Meaning scallop if the cytoplasm of the cell conforms to the shape of the surrounding cell. No, it accommodates the shape of the surrounding cell. So a blue cytoplasm, scallop, no, blue scallop cytoplasm with clump chromatin pattern. This is not, of course, myeloblast. This is not immature because of the clump chromatin pattern and the absence of nucleolus. But presence of clump chromatin, scallop, blue cytoplasm, this is most likely a variant lymphocyte. Indicative of viral infection. All right, this one, very easy. Oh, look at the immature. Why? Look at the staining of the chromatin pattern, really fine with less clumps, and the presence of nucleolus would tell us that this is an immature cell, probably a blast. And then you look at the cytoplasm. Cytoplasm is irregular, no, scallop in this side here, irregular cytoplasm. Okay, so more abundant compared to the previous, no, blast. So this is a blast, no, particularly what blast? Okay, a myeloblast. Okay, myeloblast. Look at also this area here, no, these are actually our rods, and this is indicative of a certain type of leukemia, no. We will discuss that in the next few chapters. Eyes here. Okay, look at the cell versus this cell. Is this mature or immature? This cell, these two cells, immature or mature? Immature. Why immature? Look at the light staining of the chromatin pattern and the presence of nucleolus. Okay? Light staining chromatin pattern, but presence of blue, scanty cytoplasm, very small amount of cytoplasm. It's a blast, no, with a scanty amount of blue cytoplasm compared to myeloblast. This is now a lymphoblast. Okay, lymphoblast. So your myeloblasts, these two cells, no, are very common in leukemia patients. So for myeloblasts, we have AML. For lymphoblasts, we have AL. How about this? Very easy. This one. Look at the cell. No, is these two cells, are they mature or immature? They are immature. Why immature? Look at the fine staining of the chromatin pattern. Diba? Fine staining chromatin pattern, no clumps in the nucleus. And then, look at the nucleolus. These pale and stained areas here, these are the nucleolus, telling us that this is an immature cell. And then we look at the cytoplasm. Cytoplasm contains granules. So what are again those immature cells that contains granules? Could be myelocyte or promyelocyte or metamyelocyte. But the presence of nucleoli here would actually differentiate this cell to these other types. No, this is actually a promyelocyte. No, if wala pa ni siya yung nucleoli, and then this is just the cell appearance, absence of nucleoli, then we can say that this is a myelocyte. This is a myelocyte, no, with flattened D-shaped appearance. But the presence of nucleoli here is telling us that this is a more immature cell. So this is a promyelocyte. No? The only distinguishing feature there is the presence and absence of nucleoli. Promyelocyte versus myelocyte. Which one contains nucleoli? Promyelocyte. Absence of nucleoli? Myelocyte. How about these cells? Okay, this cell. Fine staining of the cytoplasm of the nucleus with presence of nucleoli, presence of abundant granules. This is a promyelocyte, no? Nucleoli with granules. How about these cells? No? These are actually all promyelocytes. Okay? Okay. How about letter E? Uy, bilobed nucleus. Bilobed nucleus, you cannot see any thread, but it's a bilobed nucleus. You cannot see if it's segmented or not, but look at the surrounding granules. The granules are actually orange, no, red-orange in color. Most likely this is a segmented, no, segmented eosinophil. Doc, we could not see this. We could not see the segment, doc. More or less, it's kind of folded, no. But if you see a bilobe cell with surrounding red-orange granules, it's an eosinophil, no. So remember the importance of granules. Then how about these cells here? Oy, vacuoles in the cytoplasm, gray, gray, gray-violet, gray-blue cytoplasm with lacy chromatin pattern. Lacy in a sense nga, it's not darkly stained, no? It's finely stained chromatin pattern with vacuoles. This is a phagocyte, most likely a monocyte. How about this one? Presence of more or less a clump chromatin pattern with blue cytoplasm. The cytoplasm is scalloped because it conforms to the shape of the surrounding cells. Okay, this is actually scallop, no, blue cytoplasm with clump chromatin pattern. This is a mature cell, most likely a variant lymphocyte. No, variant lymphocyte. How about this cell? Oh, klaro na kayo. Bilobed with red-orange granules. This is a segmented. Look at the segment. Segmented eosinophil. How about this cell? A cell in which the nucleus is not clear. No, it's inconspicuous nucleus because it is covered with blue-black granules. Look at the blue-black granules. This is a basophil. Okay. How about this? A segmented, no, cell with fine thread, but the color of the granule is red-orange. It's a segmented eosinophil. How about letter K? Look at the fine granules here. Fine granules which appear purple or violet, lilac granules, which has segments, no, different lobes. So this is a segmented neutrophil. How about this? Basta di maklaro ang nucleus, batignaw, inconspicuous nucleus with blue-black granules. It's a basophil. Alright, we're almost done. No, we're down to our last cell line. Okay, now cells of the erythroid series. Erythroid because these cells will give rise to a mature erythrocyte or red blood cell. So this is the general ruling in the identification of the different erythroid precursors. I mentioned this already actually, no? As the cell matures, the size decreases. As the cell matures, the NC ratio also decreases, meaning the size of the nucleus decreases, whereas the cytoplasm volume increases. No, I've mentioned that already. As the cell matures, the nuclear pattern, the chromatin pattern becomes coarser or clumped compared to an immature, okay, immature cell which has a fine dispersed chromatin pattern. And then as the cell matures, it loses its nucleoli. Okay, you have to master these. And then the cytoplasm changes from blue to gray to pink. Increase of the base of a more immature cell, the more the immature the cell is, the more the immature the cell is, okay, the color of the cytoplasm is most likely blue, okay? That is because of the significant amount of RNA in the present in the different number of cellular organelles. But for red blood cell, the more mature the cell becomes, the more pink, no, or red it becomes. Actually, a mature red blood cell is termed as color is salmon pink. Salmon, no? Salmon pink. Okay? So let's start with a doc blast, another blast doc. Okay? A rubriblast, also known as pronormoblast or pro-erythroblast. Okay? So a rubriblast is an immature cell, but you look at the chromatin pattern. Okay? The chromatin pattern is very fine. Chromatin pattern with evenly stained nucleus, and the presence of nucleoli tell us that this is an immature cell. Now, how do you differentiate this with a myeloblast? So look at the picture. No, look at first myeloblast. Sige. If you can notice, a myeloblast actually has a coarser chromatin pattern compared to your rubriblast. No? Mas smooth yung chromatin pattern. But the main distinguishing feature, no, to distinguish the two is the very prominent basophilia of your rubriblast cytoplasm, tanawa, cytoplasm sa myeloblast. Much bluer, more basophilic cytoplasm of your rubriblast. This means that the cytoplasm contains more RNA material. That's it. Okay? Versus lymphoblast. Okay, look at lymphoblast. Doc, lymphoblast also has basophilia, but the amount of cytoplasm will differentiate the two. No? In lymphoblast, scanty cytoplasm, maliit lang cytoplasm, but this one has more abundant cytoplasm compared to your lymphoblast. Okay? Now, the second stage of maturation series is what we call prorubricite, also known as basophilic normoblast, also known as basophilic erythroblast. You have to know the different names for examination purposes. Now, as I've mentioned, as the cell matures, the chromatin condenses, the chromatin becomes coarser or clump, as you can see here. Diba? And the cell now being more mature does not contain any nucleolus. But Doc, what is that things there? What are these empty spaces, unstained areas in between clumps of chromatin? We call it parachromatin. Okay? Parachromatin are non-staining areas or clear areas of the nucleus. All right. And if you look at the cytoplasm, this, as I've mentioned, very prominent characteristic of immature erythroid cells are basophilia. No? Is the basophilia of cytoplasm. Okay. Next, we have rubricite. Rubricite is a small cell, no? Could be slightly bigger than an erythrocyte or a mature red blood cell. But look at the chromatin pattern. No, look at the chromatin pattern. The chromatin pattern forms very big clumps or chunks. That's why we describe here the chromatin pattern as a chunky chromatin pattern. Usually the nucleus is eccentric, no? Nucleus is eccentric, meaning it's off-centered, and the cytoplasm is very abundant and is colored gray, gray to bluish. Okay? So the gray to bluish cytoplasm denotes starting formation of hemoglobin. This is also the cell, the last the stage, no, of the erythroid precursor cells capable of cell division. Okay? So again, chunky chromatin pattern with blue to gray cytoplasm denoting hemoglobin synthesis, and the cell, erythroid cell capable, no, last cell capable of cell division. Comparing it with your metarubricite, also known as orthochromic normoblast or orthochromic erythroblast, no, you look at the cytoplasm. This is actually having a more abundant cytoplasm compared to your rubricite, no, mas gamay ang cytoplasm compared to this one, very abundant. And you actually do not see anymore the chunks of chromatin. Okay, you've seen chunky corned beef. That is what we mean by chunks, no? Bigger clumps of chromatin. You don't see that in your metarubricite because the cell now here, this is actually the last cell, no, with nucleus, is colored pycnotic. Pycnotic in a sense that the entire thing is colored dark. You could not see clumps anymore. It's colored dark with abundant blue-gray cytoplasm. Okay, this is a metarubricite. By the way, the nucleus is also eccentric. Eccentric because it's ready to be expelled, no, anytime. Then once the nucleus is expelled, now this is the first stage in the maturation without nucleus. We term it now as reticulocyte, no? The term reticulocyte is only used if we are using supravital stain to demonstrate the cell. If we are using supravital stain, we term it as reticulocyte. But if we are using right stain, we call it as polychromatophilic erythrocyte or polychromatophilic macrocyte or diffusely basophilic erythrocyte. Okay? Again, I'd like to put emphasis that we only use the term reticulocyte if the cell was stained using supravital stain, such as your gentian violet or BCB. Okay? Brilliant crescent blue. So this is how it looks like. It's a big cell, macrocyte. It's a big cell which does not contain any nucleus, and it does not contain also central area of pallor as compared to your red blood cells, which contains area of pallor. The area of pallor here is actually 1/3 of the entire red blood cell, 1/3 of the entire red blood cell diameter. No? So big cell with no central area of pallor, and the color is more or less reddish, okay, reddish to grayish, okay. The grayish there or the bluish there denotes the presence of RNA. This cell contains RNA still, okay, and you can stain the RNA, the reticulum, the RNA, through the use of a supraviolet stain. And if you are using a supraviolet staining technique, we term the cell as reticulocyte. So this is how a normal, mature erythrocyte looks like. It's a biconcave disc, okay, with a central area of pallor. Central area of pallor denotes that this area does not take up the stain. And the pink surrounding area here is actually representative of the hemoglobin content of the cell. Okay? And you know already that the function of your hemoglobin is to carry oxygen to different tissues of the body. So that ends my lecture for introduction for hematology module. Thank you very much and God bless.